The stage is all yours.
Okay, thank you. Thank you for having me here. And my name is Dewei. I'm from the Novo Nordisk Foundation Center for Protein Research and the Center for Basic Metabolic Research at the University of Copenhagen. And I'm very happy to share my PhD project, Space, Stream Proteins as Complementary Embeddings with you. And I just assume most of you knows about string. And if you don't know, string is a protein-protein interaction network, and it actually is a protein functional association network. And if you query a protein in string, you will get a network like this, and each two proteins are linked by many different sources, like we have genomic context, high throughput lab experiments, and so on and so forth. But most significantly, a lot of resources are from text mining. And we have a very good coverage of the organism, especially for many of the bacteria. And we have more than 1,000 eukaryotic species. And because we have so many users, we want to provide something that they can use, because many of them, they're not specialized in computer science, and they're not very good at coding. So we want to provide some embeddings, some features we can generate from the string networks. And you know, it's not very straightforward to get those embeddings. So because when you have a graph, you firstly you have to do the random sampling to generate the random walks, and then you get a bunch of the something like a sentence. You get a bunch of sentences, and you put them into a very simple language model called skipgram, or some very other simple models, and you can get the embeddings. And you can use the embeddings and features, and you can just do them based on your own defined labels to do classification or regression. And this method usually works very well. And I will just show you a very simple example that I'm doing with a company called DDS in Denmark. We're using the human string network embedding as features to do the disease protein prioritization. And we use the omics data to do like some very common omics analysis, where you can get the p-values of each protein from the human proteome. And then you probably have some proteins that are differentially expressed in different people. Some people are healthy, some people get some diseased. And then we just use a logistic regression model to do a binary classification. And this is outperformed the traditional omics analysis. And if you are interested in this project, you are also very welcome to our poster on Thursday, D460. But also, there are a lot of limitations of using traditional random walk-based, like the no-to-vac embeddings. Like we have two networks, for example, human and mouse, and we both have the actin proteins in us and mouse, because we are very similar. And actually, those proteins they have very similar functions, and they also interact with other proteins that are having some similar functions in both us and mouse. But still, if you use no-to-vac or some other network embedding method, you can get the embeddings, but they are not comparable. Like here, I just show you a very naive distribution, like the actin protein in mouse and human. So what we want to solve that we want to create some embeddings that are comparable across different species, but we only focus on the eukaryotic species. And we also think that the embeddings from the network that are complementary to the sequence embeddings. So we provide both the network embeddings and the sequence embeddings. And then the way to do it, that we can use we call the embedding alignment based on the oslogs. And the oslogs are like protein. They have similar functions, but they are in different species. But if you look at the evolution history of any true proteins, like actin protein in human and mouse, they evolved from the same common ancestor. And even after some speciation, they still have very similar functions. And we call those proteins as oslogs. And you can get this kind of oslogs from a database called Eggnog. And then I will just show you how we do the embedding alignment. So imagine we have those embeddings of human mouse with Node2Vec, but their embeddings are completely independent. But still, we know that some proteins they might get together because they are in the same functional module, or they share the same pathways. And then we when we do the embedding alignment, we just put those oslogs into the same cluster to make their embeddings as close as possible. But at the same time, we don't want to change the information that we captured by the original embeddings. And we do this through a way called thread coder. So firstly, we have those protein networks in string, and we just use Node2Vec to create the embeddings for each species. And this step, they are completely independent. But then we use the autoencoders, and we for each of the species, we have one autoencoder, and the encoder would project the Node2Vec embedding for each species into the latent space, and the decoder would reconstruct those latent space into the input. And we have two loss functions here, and of course we have the reconstruction loss as most of the autoencoders do, but we also have the alignment loss, where we use a loss function to push those oslogs as close as possible. And here we could use any kind of distance matrix like the Euclidean distance or cosine similarity. But in practice, we just find that the Euclidean distance works best for us. But at the same time, we still got a very big issue with this kind of method. Like we have more than 1,000 eukaryotic species, and many of them they are not well studied. But we still have some model species that are well studied. So we don't want to distort those model species by aligning them all together with those not well studied species. So the way we do it that we firstly selected some seed species that are very well studied, and most of them they are model species, and we select them across different 4 kingdoms. We also try to balance the number of species we selected on each kingdom. And then firstly, we selected 48 species, and we know that they are well studied. They also have very good network quality, and we did some benchmark. And then we just do the embedding alignment on those 48 seed species. And then for each all the other non-seed species, we just align the embeddings of them into the 48 seed species. We fix the embeddings of the 48 seed species and push them all together into the aligned latent space. And then of course, we do reconstruction to keep the original information captured by Node2Vec. And then we can check if the embeddings are actually aligned. We just use a UMAP and to do the dimensional reduction. And then in the 2-dimensional space, we can observe if the network distribution of different species are similar or not. And here we selected 5 species: human, yeast, Arabidopsis, slime mold, and rat. And rat is from the non-seed species. And firstly, if we compare human and rat, because we are very close in the phylogenetic tree, so we definitely share a lot of the similar patterns. But of course, there are some unique proteins in rat, of course. And if we compare other species to Arabidopsis, and you can find on the left side, there are a lot of the unique proteins, and many of them are from plants. And then we just did a quality control with click pathways. So we want to know if after alignment, our embeddings are better or worse than the original Node2Vec embeddings. As you know, string covers a lot of the pathways, so it should perform very well on the click pathway quality control. And we also take the Protein-5 sequence-based embeddings, because we know that our network-based embeddings should be much better than the sequence embeddings, because we have the network information and the sequence embeddings don't have. So firstly, we selected 12 species from different kingdoms, but I guess you cannot see the text here. But we selected species from 4 different kingdoms. In general, you can notice that the aligning embeddings, the solid lines, they are very comparable to the node-to-vec embeddings. And they are all so much better than the Protein-5 sequence embeddings. So that's why we say that our embeddings are complementary to the protein language models generated sequence embeddings. And then we did a downstream task analysis. We use our embeddings to predict the subcellular localization, just to use a logistic regression model, because if you get a very nice embedding, you don't need a very complex model. You should use a very simple model. And here in this task, we have 10 labels or 10 localizations. And firstly, we just did a UMAP, and it's a sanity check to know if our embeddings can capture some localization information. And here we also introduced a new terminology. We call it the space embedding, where we concatenate the Protein-5 and the align embeddings together. And if you look at those precision recall curves, you can notice that our space embeddings just outperformed the Protein-5 embeddings, because we offer the information from the network. And then if you go to check each of the localization, you can find that actually in many of the localizations, we cannot improve the prediction very much, but actually we always increase the prediction. And especially in lysosome, Golgi apparatus, and peroxisome, our aligning embeddings are just so much better than the Protein-5 sequence embeddings. And of course, if you concatenate them together, you can get even better results. And then we did a protein function prediction, and also with logistic regression. And here we have 3 aspects. We have cellular component, which is another way of doing subcellular localization, but it has more terms to predict there, and the biological process and molecular function. And if we look at the cellular component, of course, we're better, because we're better at a subcellular localization prediction. And in biological process, you can notice that the align embeddings, the black line, is already better than the Protein-5 sequence embeddings, because we are providing the network-based information. And if you just concatenate them together, you can get even better results. But in molecular function prediction, why we cannot get further improvement is because of that we already have the function information inside the sequence, because the function is defined by the motifs and the domains. And finally, we have a preprint. You can check all the results we have here. And we just public our embeddings, release our embeddings, the network embeddings, and the Protein-5 sequence embeddings on screen. And we also have a GitHub repo to show our implementation and some other documents there. And finally, I would like to thank my supervisor, Lars, and he's there. And he's now a director at LeadS, and also he's a YouTube influencer, and he has a lot of the he has a lot of the YouTube videos about string. And if you have any questions, you can also watch his videos. And also very thanks to all the ESN group members and our collaborators at the Swiss Institute of Bioinformatics, Dami and Christian, and also the collaborators at LeadS, Annalisa, Julia, Clara, Daniel, and Rasmus. And finally, I would like to stress that this project is supported by Lumi, which really give me a lot of the help in this project. Thanks, and I'm happy to take any questions.
have a question about very interesting talk. I have a question about the alignment loss. What is the difference between the alignment loss and the contrastive loss? And another question is like you said, like you from your map decided you have to have another method to quantify the alignment.
Yes. So the alignment loss is that we just push the oslogs, the embeddings of oslogs as close as possible. And the reconstruction is that we try to maintain the information during the alignment. And to answer the second question, we actually did a lot of the benchmark to see if the proteins are aligned. So we actually check the distance distribution of the oslog pairs and the non-oslog pairs. And of course, the distance of the non-oslog pairs, they are way closer than non-oslogs.
Hi, I have a question about paralogs. Paralogs are orthologs within the same species. You probably know that, but I'm not sure that everyone does. So, how does your system works when you have paralogs in the same species and when new paralogs are appearing in when you are comparing the various orthologs?
Yes. We do have some paralogs here. So your question is like sometimes we might have some like let's say 10 proteins from mouse, but only one protein from human, and they are all oslogs, right? And we weight the oslog pairs based on the number of the proteins they have. So if we have 10 proteins from mouse, and each pair they're weighted by 0.1.
And when you try to do functional predictions, can your method capture the different functions, the slightly different functions which each paralog have?
That's a good question. We didn't actually check how it performs on different proteins.
I can shout pretty loud. So I have a question about the embedding process itself. How do you sort of optimize the depth and breadth searching? Have you done some work to figure out the best search strategy for the embeddings?
You mean the node-to-vec embeddings?
The node-to-vec part, yeah.
Yes. So we did hyperparameter tuning. So you can use either breadth first or the depth first with search. And we try to use the embeddings to make link prediction. So we partition the network into different links, and we use the training set to predict the link invalidation and test sets. And we find that actually either the breadth search or the depth search doesn't matter that much. So you can get very good performance in link prediction with those two kinds of ways.
Okay.
So we do have online questions.
Yes.
So from I don't know. Sorry, I cannot read it. So the question. Thank you for the excellent talk. I actually read the research paper for space, and I was excited to see from a new perspective. Could you elaborate on why you choose not to back and profile for the embedding over other similar models?
Yes. So there are a lot of methods you can choose, and in general, we also look at some papers, and in general, the unsupervised models like node-to-vec, they are much better than the graph neural networks. And out of those unsupervised models, node-to-vec they are more robust in general. Like you might have some very unique or novel method that can outperform node-to-vec, but in general, we just use a method that works in general. It's not based on any matrix.
Okay, and a quick question from Claudia: do you plan to include tissue specificity in manual species?
No, we didn't. We could try. Yeah, thank you.
So where is our next speaker?
Thank you.